Hello, everyone. I am Deepthi, and I work as a scientific curator in PRIDE's team. And my topic today is about PRIDE databases, the proteomic data submission, access, and visualization. So just to make sure that this is going to be very basic tutorial about the whole process, and I'm going to talk about little about the PRIDE submission process and our web page. So PRIDE, we basically have two main aims, is to archive all types of mass spectrometry based proteomics data, and to also to support the reproducibility of the research data, and also to enable the reuse of these data produced by the researchers. And the second aim is to disseminate those mass spectrometry data to other bioinformatics added value resources within EBI, or sometimes even outside of EBI. So as for today's talk, I'm mainly going to focus on the PRIDE archive and the submission process. So just to give you the very little background about the proteomics, proteomics is basically the study of a proteins in a time and space, or protein interaction, post-translation modifications, and also the signal transduction. It can also be the study of signal transduction, protein localization, or protein turnovers. And about the basic approach, the main approach is mass spectrometry. There are many other approaches, but the main approach is mass spectrometry, which is widely used by the researchers. And just to give you the basic, very basic overview of the process, is that here you have the cells or tissues where the proteins are extracted, and then they are digested into the fragments, mainly by the enzyme trypsin, and then these peptides are separated by liquid chromatography, and then they send into the mass spectrometer, and then the spectra is generated and then analyzed later by the researchers. So just to focus first on the data submission process, first I'll tell you a little more about the PRIDE, that it stores, archives basically mass spectrometry based proteomic data set, basically mass spectra data, raw data, peak list, and the other files. And also all proteomics approaches are supported now. PRIDE is one of the core data resource of Elixir. And of course, we don't work in isolation. We have five more, five more sister repositories: JPost, iProx, Massive Peptide, and Panorama. But PRIDE is the main leading and the founding member of ProteomeXchange consortium. So in till May, we have approximately or more than 27,000 datasets stored in PRIDE. And you can see from the past couple of years, the amount has increased almost double, and it is like increasing in 500 dataset on an average. And PRIDE has and these datasets are coming from more than 15,000 research laboratory, and we constitute among all ProteomeXchange dataset, we constitute approximately 82% of the datasets. And the most, according to our PRIDE statistics, the most studied organism is human datasets among more than 3,000 taxonomies. So we are not only increasing in volume, we are also being the data from PRIDE has been widely reused, and you can see from this chart that the data download has been increased immensely. Apologies that we don't have the last year data, last year download data because of technical issues, but it is only increasing every year. So this is just the basic workflow that scientists generate their data by using mass spectrometer or any other approach, and they upload their data to PRIDE, and one of the curators check the process, and then the data being submitted to PRIDE, and then an accession number being generated for them to use into the manuscript for the publication purposes. So PRIDE is basically a data submission driven resource, and it aimed to store the author's original view of the data, and it also captures the reanalysis of those public data sets which we promote widely, and it stores the identification results of protein peptides, including post-translation modifications, and it also stores the mass spectrometry output files, both raw and peak list files, and protein and peptide expression values, additional technical and biological metadata, and if they have other files like gel images, scripts, etc. So, and the PRIDE accept mainly data sets via PX submission tool, which is a desktop application submission tool. It is an open source and standalone tool, and you can annotate your data set with the tool, and also link your files and upload it to PRIDE. It is based on the Aspera file transfer protocol, which makes it super fast, and terabytes of data sets can be submitted in a day or two. So the required files for to upload your data to PRIDE are the mandatory files if you are doing different types of submission, complete or partial submission. I'm going to talk about it later. So the basic files are raw files, which is basically output of mass spectrometry instruments, and it can be in a different format, mainly two different format, MZML or MZXML. Then a results file in case of complete submission, MZ-IDEntiML or MZ-TABs are supported. Peak list files, it is mandatory in case of complete submission, and then metadata are sufficient, like additional details of the of the samples, and then SDRF, which is sample to data file format. It's a new, but right now it's optional, but we recommended submitters to provide that. So I will, before starting to the live demonstration, I will tell you a little more detail about the SDRF, because it is quite new format which we promote users to use. So for till now, recently PRIDE only used to capture very basic information, metadata of the sample, but just the very basic, like title of the title, publication, instrument, or very basic detail. But the structured metadata with the individual samples were missing, which was making it really hard to annotate for the purpose of reuses. So there was a need for a specific proteomics file format, which promotes the mass spectrometry based proteomic data, which accommodates them. So a new MAGE-TAB for proteomics has been proposed, which is basically an extension of the MAGE-TAB, which was already used by transcriptomics. So MAGE-TAB for proteomics is consists two file formats, IDFs and SDRFs. So IDFs are basically the submission PX file, which we already store during the submission process. But the SDRF is a new file which we ask submitters to create which can accommodate the SDRF proteomics experiments. It has a new flavor of SDRF proteomics, and it also can support the proteomics experimental approaches. So if you really want to read more about the SDRF file formats, you can go to the GitHub page here, and there you can find that there is additional documents where you can read through, and we have a lot of annotated projects already, which was done by the team or the collaborators, and also the templates are there. For example, if you have the sample data sets, or plants, or cancer cell line, or even default, you can select and start annotating your data sets. So till now, right now, we have more than 200 data sets which has already been annotated in GitHub. So now I will switch to the live demonstration of PX tool. So this is how you will get a link from from Pride web page, from where you can download the latest PX tool, and when you launch it, you will see the login window where you can enter your Pride credentials. But yes, before using the PX tool, you must register with the Pride first, and then you enter those credentials to the PX tool. Yeah. So this is the step 1, where you have to decide to go for complete submission or partial submissions. So we always recommend to do a complete submission, because you can provide your identification results in MAGE-TAB or MAGE-TAB, and this complete submission, it can be fully integrated with the results in a Pride, and you can also visualize them. And only drawback with the partial submissions are that they cannot be completely integrated for with the Pride results. So next window, the prerequisite, they're asking that you must be having result file, raw file, lab head and experimental details in your hand. So yeah, from here you can enter the metadata related with your project. So I'm just going to copy paste some of the details here. Give me a second. So this is the title of my project, and you can enter the keyword. It can be anything like based on your data set, and the description. Yes, and then sample processing, the project description, and then sample processing protocol. So there should not be any space, yes. And then, yeah, I'm just randomly pasting data processing protocol. And then here you can select the experiment type. For example, here I'm selecting the shotgun proteomics, and then you click to next. And here you can upload your actual data. So I'm going to upload the test files which I have prepared. So yeah, so here I have MZ-ID, which is my result file, raw file, and the peak list. And also I have already annotated my SDRF, so I can upload it here. So by default, PX tool tags them according to their extension. But since I have just one result and one raw and one peak list file, it's easy for me. But if you have more, then it's very important you check before you proceed to next step that all the files are tagged properly. Then you go to next step. And yeah, you have to give it a few seconds. So here it is asking if you want to calculate the checksum. So we have recently introduced the checksum because it's very important that we receive the same file which submitters have uploaded. So I click yes because it's important. So I go back and I go to my PX working place where I have the checksum, and then you choose it and go to, because it is automatically created for you in your PX tool folder when you launch the PX tool. So you just have to wait now, and then it will lead you to next window. So sometimes it takes a little while. Yes, okay. So yeah, now it's calculating because now it's realized that I have the checksum file. It's 100% complete, so I can click next. So here, since I have only one result file, I don't need to add the relation or associate those raw and peak list to my result. But if you have more than one result file and you have different raw files associated with each result, so you must define with each result file. So here, because it was just single result file, so it was automatically selected. So I can skip this, and then I go to next. And here you have to annotate your result file, your sample. So if you click to in the annotate tab, you will see that you have the species, tissue, instrument, modification, if any, cell type, disease, and the quantification methods. So here, for example, I choose Homo sapiens. Yeah. If you cannot find, generally we have most of the species or any metadata in our drop down list, but if you cannot find them, you can always select other species and it will connect you to ontology, our OLS, which where you can type and find your particular sample, particular metadata. And then for the tissue, I'm selecting blood, and yeah, any. And then for modification, you can select if you know, or if you have, for example, coagulation or oxidation, or if you don't want, or if you don't have, you can also select no PTM, but in that case you have to remove this. And then cell type, disease, if any, and then the labels, quantification method, or label free. Here it's recommended to provide the experimental factor, but you can also skip if you don't want to. Like I'm going to provide here biological replicates, for example, 2. So yeah. So I click to, yeah, so next, you can see all the metadata which I have provided here in here. And if you find that something is wrong with selected, you can always remove and go back and select next time. So if you click next, here you can provide your PI or scientist. For example, here I'm going to provide the student supervisor, then email so at Okay, and then affiliation. Yes, so you can go to next step, and yeah. So here in this step, if your project comes under larger projects, like if you have any parent project, for example, human proteome, or the European data sets. So we have some here, human proteome project or whatever. So if you can find it here, if you have any, you can select it here, which help them to group with those data sets. And if your data set is already published, or you can provide the PubMed ID also here. And if it is a reanalysis of any public data set, you can mention it here. And this is to add if you have other omic link, if your data set is submitted into other repositories like transcriptomic or any other, so you can always provide the accession or the link at this stage. And then at this step, you can see that I have 5 files, which is where one peak file, result file, no search file because it's a complete summation. I can provide search file, but I didn't have it. So, and then raw files and one other file, which is my checksum file. So it looks okay. So I should accept, I have to accept the PRIDE data policy and archival, the license. And then I go to submit. And here it will ask you to export the submission PX file. And I always recommend submitters to do that in case if their if PX tool crashes for whatever reason, any other reason, you can always have your submission PX file. And if you cannot use the tool, you can always ask the PRIDE support, and you have to provide, in any case, submission PX file and the checksum file to us with your data sets. So, and also you can check your submission PX file, and you can send us sometime if you want any kind of editing, any kind of modification in your metadata. You can update your submission PX file and send it to us. So it's a good practice to export your submission PX. And then your hit the submit button and your data set is submitted. Because I'm in the training mode, you don't see any green bar over here. So, but you will be given the submission reference ticket, which you can use to if you find that you need to ask us anything regarding your data set before the submission. You can always reference your ticket number and ask us anything. So this is not the accession number. Accession number will be only provided once your data set is submitted. So from here, the next step would be to we have this semi-automatic pipeline where your data set will be picked and validated. And if it pass the validation stage, the curator will check the report and submit it to the PRIDE. Launch the submission pipeline, and it will be submitted, and you will be given an accession number. So by clicking this, you can give your feedback as well for the tool. And yeah, this is the tool. Now I'll switch back to my presentation. If you have any question regarding the tool, you can always ask PRIDE support, and we'll be able to answer it. So I just keep it as a backup if anything happens. So yeah, I have covered these steps. So now I will focus on how you can access and visualize the PRIDE datasets. So this is the main landing page of PRIDE web, and you'll see that we have 5 resources like PRIDE Archive, PRIDE Active Archive Annotated, Active Spectra, Archive Spectra, Spectral Libraries, and Peptidome. And then tools. This is the tool tab where you can download your submission tool or inspector tool suite or utility libraries. And the doc page where it says doc, but actually it's a help page where you can read through. We have very like we have provided a very explained detail of the whole submission process, the type of file you need, the mandatory files, and everything, troubleshooting, everything is present here. So you can always go here and read according to your need. And here you can register before submitting your data set. You must register here. And yeah, with the if you want to search anything, you can provide in the search box your search key. If you want to data set or cancer, human, or stress, tissues, whatever you have, you can search here. And these are the tabs for the resources like Pride Archive, Pride Peptidome, and Spectra Archive. So once, if you, for example, I've given the stress keyword, and I was, the result shows like there are 1,200, almost 1,300 data sets related to this search. And then, so you can also specify the date, like, for example, you want data sets from 2019, not very old. So you can even add additional searches. And then you can sort your results according to the field in this drop from here, this drop down. And you can also order the results. And if it gives you, like in my case, I have got almost 1,300, so you can scroll over the pages. So once you got your data of interest, you click there and you can see that this is the project accession. These are the project metadata field, and these are the project file, which I'm going to zoom in more. And yeah, so this is basically the metadata which you have provided, like which you saw me providing during my upload. So these are the title, description, sample, and data processing protocols. So all you can find it here. Because it is a public data set, you can see also that when it was submitted and when it was published. And then this is the paper in PubMed. So we have the links over here. And these are the other metadata of the project, like organism Homo sapiens, modifications, instrument used in the experiment. And if it is a complete submission, you can, as I mentioned before also, that you can, the benefit of having complete submission is you can visualize and inspect your peptide and protein evidences with spectra identified in search in each data set. So if you go click there, you can find these screens in the next page, and you will see that the protein groups, peptides, hits, PSMs, and if you select any, you will see the sequence coverage and spectra also. So that this is the benefit of having a complete submission in PRIDE. And if you go to the project file section, you can find all the files which you can scroll over. So these are the, like, you can also, if you want to download all the files, you can go to the project FTP and you can download from there. Or if you want individual files to download, you can have it here. Each file has its own tab which you can click and download. So this is like, now I'm going to end my talk. You can always revisit the PRIDE website. You can read through our tutorials, our web help, and also we have another online very short quick tour about PRIDE. You can always go through. And at the end, I would like to thank my team, my manager, Juan Antonio Vizcaino, and our developer teams, and our bioinformatics and data curation team. and of course our funders and other collaborators. So I would like to thank you all for listening, and now we can go ahead with the question.
Thank you. Thanks a lot, Deepti. Thank you for webinar today. So if you have any question, please type them in. While we are waiting for questions, is there any general comment, Deepti, or anything new coming up for Pride you would like to share?
In future, we are planning to do more of these kind of webinars and more like for the SDRF, because it's new format which we have proposed. So we might do in future a very specific webinar on only on that. So if you want to know more, I always welcome. You can go to GitHub page in anyways, and if you are interested, you can also select any public proteomic data set and you can annotate out of interest if you want, and you can actually push into the GitHub, and we can review it, and yeah, it can be in GitHub as yeah. So if you are interested, you can always do that, and you can always ask questions there as well.
Okay, so I think we have one question in now. So hi, thanks for sharing. Does the SDRF cover linking raw data to patients? For example, patient age, gender, overall survival time, treatments, etc.
Yes, because I didn't actually show you the exact SDRF file in very detail, which I wanted to, but yeah. So it has the columns where you can have your sample and age, or like whatever you want to annotate, you can do it, unless until you're not providing the actual name of the patient. Everything you can annotate there. And you can always select, as I mentioned, you can select by trying yourself. You can annotate one of the data set and push it to GitHub to see that how it goes, and before starting your actual annotation with annotation of your data set.
All right, so we have next question in. What is the most efficient way to search in the database?
From Pride Web, you can search from Pride Web, as I showed there, that you can enter your keyword. And also we have our, it's again new, and we are planning to do that webinar as well about how to search and download the data sets from Pride. So it's like our API, it's a RESTful API. You can even find the link in Pride Web. And you can, it has the query format where you can query our database directly rather than going to web page. You can directly query our database. So you can find the link there. And if you want, I can provide it to you later, that RESTful API thing. So you can query actually our database, and that will give you the very accurate results.
Okay, so we have another question now. Thanks for the great talk. Is there a specific place to store workflows? For example, Proteome Discover workflows. Those settings are often crucial to reproduce a previous analysis.
Yes, actually the upload process, submission process is same for all the approaches. But as you mentioned, the PD, if you are confused, you can always go to our doc page, and there you can find that if you have a very specific approach and you don't know how to like upload your files from that approach, you can always read through from there. So yes, you can do that, and it's the same process for everything.
All right. Will the SDRF annotated datasets be updated only in GitHub, or will it also be updated on Pride?
It is already updated on Pride. As I mentioned, there was in resources, you see that annotated, what was that? But you can check, and even if we have that, whatever is in GitHub or whatever, if users provide, they provide with when their dataset is public, and if it is in correct format, SDRF is a correct format, it will appear with their dataset, which makes it really special because you can see all the very structured metadata information. So yes, you can find from Pride Web as well. We want SDRF to be used more widely, and we are moving in that direction. Right now in PX tool, it's optional to provide, and we even ask submitters, if they cannot annotate in a correct format, at least they should just upload it as a other, not with the experimental design format, but other, and then we can in future take it and annotate it properly, and then it will be shown with their data sets in Pride Web. So right now we are in a very initial phase, but we really want it to move forward.
Deepti, thank you again for the webinar today.